Tumors can avoid immune response by adopting some escape mechanisms. Unlike infections, tumors rarely produce danger signals and thus do not directly activate the innate immune system through any receptor or pattern recognition molecules. This means that the innate immune response normally does not attempt to kill tumor cells, nor does it alert adaptive immune response about the presence of the danger. Well, this is not the fault of immune system, except in immunocompromised patients, but tumor itself develops such mechanisms so that it can avoid immune response. These set of mechanisms are called escape mechanisms of tumor cells. So, let's see what can be different escape mechanisms of tumor cells. There are a number of escape mechanisms that are responsible for the absence of an effective immune response against tumors. Let's try to understand some important mechanisms one by one. Starting with avoidance of recognition of tumor cells by the host immune system. This avoidance of recognition can be achieved by lacking tumor peptides. Each autologous protein is degraded within the cytoplasm to form peptides, consisting of 9 to 12 amino acids. These peptides are then conveyed by a transporter system called transporter associated with antigen processing, or TAP, to the endoplasmic reticulum, where they bound by class I MHC molecules and presented on the cell surface to cytotoxic T cells. But in case of tumor cells, they may not form such tumor peptides that fit into the binding sites of patients' MHC molecules, and so they avoid detection by T cells. Some tumor cells also avoid recognition due to defective antigen processing machinery. As we mentioned, this TAP, that is transporter of antigen processing, further process the antigen. But tumor cells may become deficient of this machinery, that is, TAP deficiency. And so tumor peptides are not transported to endoplasmic reticulum. Thus, no binding with MHC, and ultimately, no detection by immune cells. Other than this, many tumor cells even lack NAHC class I molecules on their surface. And the possible reason of this absence is the downregulation of MHC genes. As a consequence, the tumor antigens are no longer able to bind with MHC. Thus, there will be no antigen presentation on the surface of tumor cells, and therefore no recognition by cytotoxic T cells. So, in a nutshell, we can say, tumor cells avoid recognition by the host immune system by lacking tumor peptides, so no peptide, no tumor antigen, thus no recognition, or by defective or no processing machinery, so no TAP, no peptides processing, thus no recognition, and lastly, by downregulation of MHC on no MHC molecule, no antigen presentation, thus no recognition of tumor. Now the second escape mechanism that tumor cells adopt is by their immuno-regulatory effect on leukocyte functions. Tumor cells first affect leukocyte function by secretion of immunosuppressants. Tumor cells secrete a wide range of substances known as tumor-derived soluble factors, TDSS, which decrease or inhibit the local immune response. These include cytokines such as interleukin-6 and interleukin-10, as well as tumor growth factor TGF-beta and vascular endothelial growth factor VEGF. Such production of immunosuppressants by tumor cells is also called as subversion of immune response by tumor cells. Another way by which tumor cells affect leukocyte function is by shifting balance from Th1 to Th2. Tumors often have a potential of shifting the balance from Th1 to Th2, where Th1 type of immune response is characterized by certain cytokines, like interferon gamma, that generates inflammatory responses. By contrast, Th2 type of immune response is characterized by cytokines like interleukin-4 and interleukin-5 that tend to produce anti-inflammatory response. So when we say tumors have potential to shift balance from Th1 to Th2 type of immune response, it means that Th2 anti-inflammatory response counteracts the Th1-mediated anti-tumor inflammatory processes. Now, third effect is by receptor blocking of immune cells. Tumor cells, together with cytokines and chemokines, also secrete some soluble receptors. And these soluble receptors then induce apoptosis of effector cells, such as human leukocyte antigen 1, HLA1, as well as other soluble receptors bind to the surface structures of effector cells, like cytotoxic T lymphocytes and natural killer cells, and thus block their receptors. This receptor blockage prevents recognition of tumor cells and activation of further lymphocytes. So, the presence of these soluble substances decreases the activation of effector cells. To sum it up, we can say, tumor cells escape immune response by altering the function of immune effector cells, and it can do so by secreting some immunosuppressants, or by shifting balance from T1 immune responses to Th2 tumor-favorable anti-inflammatory responses. And lastly, by blocking receptors of effector cells. In this way, it alters immune cells themselves and thus decrease their activity. Another escape mechanism is linked with relation between myeloid cells and tumor development. Suppressive cells outside the T lymphocyte system are sometimes collectively referred to as myeloid-derived suppressor cells, MDSCs. These MDSCs are the heterogeneous population of cells consisting of immature cells of myeloid lineage that are macrophages, granulocytes, dendritic cells, and other cells in early stages of differentiation. And the formation of these MDSCs is induced by substances produced by tumor cells, such as GM-CSF and VEGF, etc. These substances also act as chemoattractants and attract these undifferentiated MDSCs from bone marrow towards tumor site. Now these MDSCs undergo functional and biochemical modulation in the tumor environment, becoming tumor-associated MDSCs, like tumor-associated dendritic cells and tumor-associated macrophages. These immature dendritic cells not only do not initiate an antitumor response involving T lymphocytes, but are actually inhibitors of such a response, causing a state of peripheral tolerance for tumor cells. These MDSCs also migrate to other sites and inhibit the activation of tumor-specific T lymphocytes. In addition, MDSCs inhibit the immune response to tumor-associated antigen by blocking anti-tumor T lymphocytes. Furthermore, MDSCs produce nitric oxide synthase, leading to the generation of nitric oxide, which has immunosuppressive effect. Other than MDSCs, another relation that aids in escaping immune response is between T-regulatory, Treg, lymphocytes, and tumor development. These regulatory T cells, Tregs, are a specialized subpopulation of T cells that act to suppress immune response, thereby maintaining homeostasis and self-tolerance. It is its positive role. As normally, these regulatory cells have an important role in regulation of certain antimicrobial defense mechanisms. Also, they are part of the mechanism inhibiting selectively activated cells with autoreactive properties. This prevents the development of autoimmune processes caused by an excessive immune response to self-antigens. But under certain conditions, regulatory T lymphocytes may exert a negative effect, inhibiting the antitumor immune response. Elevated numbers of Treg cells have been observed within the tumor mass in close proximity to the tumor. And this increased frequency of Treg cells cause problem by suppressing immune cells functioning and inhibiting anti-tumor immune response. Another escape mechanism of tumor cells is through apoptosis of incompetent cells. The cells of some tumors are capable of inducing apoptosis in immune effector cells. For example, melanoma cells can release exosomes that contains Fas ligand, FASL, which interacts with the Fas molecule on the surface of lymphocytes, leading to their death by apoptosis. Here, exosomes are small particles of 80 to 200 nanometers in diameter, whose potential role is to transmit signals among antigen-presenting cells. The Fas molecule belongs to the family of death receptors, which are involved in initiating a cascade of apoptotic signals in T cells. In the initial stages of the neoplastic process, tumor cells secrete exosomes containing FASL on their surface and cause apoptosis of T lymphocytes. Mechanism of apoptosis is somehow essential for homeostasis, as it efficiently eliminates superfluous cells. But numerous mutations accompanying tumor formation lead to a blockade of the intrinsic apoptosis pathway, and in consequence, prevent the effective elimination of neoplastically transformed cells. And so the overaccumulation of dead cells disturb homeostasis and also can cause mass formation. Now lastly, tumor cells escape immune system by utilizing immune checkpoints. Under physiological conditions, the immunological pathways are functioning properly, which plays a crucial role in regulating immune responses to pathogens and preventing autoimmune reactions. These pathways are regulated by immune checkpoints, which under malignant conditions may be used by tumor cells to facilitate their growth and progression. Example of one of these checkpoints is programmed cell death protein 1 or PD-1 protein, also known as CD279, which is a cell surface receptor of T cells and pro cells. PD-1 binds two ligands, PD-AL1 and PD-L2. In order to evade anti-tumor immune response, tumors can express PD-L1 and PD-L2 ligands. Interactions of PD-1 with any of these ligands cause chronic T cells inhibition, which leads to their anergy and decreases immune response to cancer cells. So this was all about escape mechanisms of tumor cells that they adopt to avoid immune responses. Let's revise them quickly. Tumor cells want to be protected from host's immune response so they can fully grow and develop. For that, they need to escape from host defense by certain mechanisms. So they do that by avoiding recognition by immune cells. That can be achieved by lacking tumor antigen or by lacking processing machinery, or even by lacking presenter molecule, that is MHC. Another way to escape that they adopt is by affecting leukocyte functions, by either secreting immunosuppressants or generating an anti-inflammatory response, or by receptor blocking. Another escape mechanism is increasing production and recruitment of myeloid-derived suppressor cells by tumor cells, and by increasing concentration of T regulatory or TREG lymphocytes by tumor cells. Tumor cells can also escape immune system by promoting the apoptosis of effector cells, and by decreasing apoptosis of tumor themselves. And lastly, they avoid immunity by utilizing immune checkpoints for their own growth and progression. Despite of all these escape mechanisms adopted by tumor cells, and immunologists have hoped that by appropriate stimulation of stronger innate or specific immunity, vaccination, the immune system could contribute to the eradication of cancer. In the last few years, the enormous effort devoted to this problem has begun to be translated into some clinical successes that we will discuss in next section under immunotherapy.